Me? Hello? Yes? All right. I see Ned heads nodding. So hi, everyone. My name is Guillaume, and I'll be presenting this last presentation for this last session on what I call CRISPR states and genome editing as a biopolitical means. So what we've been talking about for those two sessions is how much CRISPR is both a landmark discovery in the life sciences, but also an ubiquitous slogan that we find in almost all biotech spaces. And this CRISPR slogan has become a very fertile abstraction to the extent that it has managed to concatenate very large arrays of biotechnological dreams, nightmares, maybe promises under one single very large banner. So we've heard scientists talking about controlling Homo sapiens' evolution. We talked about gene drives, so wiping out entire species, adapting plants with GMOs or gene-edited plants to the upcoming dystopian climate of the Anthropocene, et cetera, et cetera. So we have this thaumaturgic well that is nearly bottomless with CRISPR. Now, all those concerns, the political concerns that are being raised with a capacity to alter genes in living organisms are not especially new. We could make a history of that. Maybe you've read the recent book by Françoise Baylis, which she does that. She does a small history of those concerns. But the most relevant summary of the political component of those concerns, I believe, has been made even before CRISPR, more than a decade ago, by Sheila Jasanoff. In her book, Designs on Nature, she asks this in the introduction, I believe. She asks this twofold question: will continued advances in the life sciences produce a new genetic underclass? And will they simultaneously increase the state's already immense power to define, classify, and regulate life itself? It should be noted that at the end of that sentence, Jasanoff herself writes a note that sends back to La volonté de savoir de Michel Foucault. So it's a clear allusion to biopower and the state's power to regulate life, le vivant. So this presentation has two main arguments. Is to say that with CRISPR, what has changed is that the first part of the question has been answered, and that the second part, that of the role of the state and its power to define, classify, and regulate life itself, is not being asked anymore, or at least is not being asked with the same intensity that it has been asked before with similar technologies. So for the first part of the question, well, I'll get that out of the way pretty fast. As you know, and we've reminded everyone here in the previous presentations as well, genome-edited babies have been born, at least 3 through 2 pregnancies, maybe more than 3, we don't know that. Oils from extracted from TALEN-modified soybeans, so TALENs that are programmable nucleases that came before CRISPR, but now everyone is working with CRISPR as well, are already in the market in the US. And hornless cattle have been brought to life and then exterminated following an FDA injunction because they had genes from the plasmid that had not been selected for in their screening. And we learned very recently that Cosmo, the first of the second generation of genome-edited cows, had been born. It's a work led by UC Davis. And we learned that this Cosmo, this little calf, edited through CRISPR, had a 7-fold inclusion of the target variant in its 17th chromosome. So that's interesting to see that HDR in large mammals, even small mammals, it has been shown in mouse before, but even in large mammals, it's a mess and it's not really working very well. So that's out of the way. We can see right now that the biosphere is home to both the edited and the non-edited. I'll let you decide which is the underclass and which is the other one. But the question of the state's power using CRISPR is not really being asked anymore. And this is a question I'd like to focus on in this presentation. We have a profusion of biopolitical, biopower theories that have all their own complexities, but it seems that bioethics has been leading the debates on what should or should not be done with CRISPR. And that might have diluted the discussions on what genome editing meant for the state powers on life. So this presentation, depending on the time I have, will try to deal with three parts. So first, I will show the data I've been working on to make the point that most of the political controversies mobilizing CRISPR are not really specific to CRISPR. So all the arguments, debates, and actors that we find in today's CRISPR controversies have been mobilized before in the GMO controversies, in PGT, pre-implantation genetic diagnosis, and MRT, mitochondrial replacement therapy. But I will show as well that what's different and what is new with this new iteration of those parent controversies is that the intensity of the requests for public engagement and social consensus is both very high. But then whenever it happens, and I will take the example of France, in which we had this bioethics law revision that was accompanied by an exercise of public consultation, a very large one. This exercise has been denounced by its own organizers, saying it's not representative of what the French public opinion or general public, whatever that means, really was like. The second part, I'll show how we can try to get out of this mess, how this collective inability to try to decide what is at stake with this genome editing power for the state. And so who is the we, who is the community that has to discuss genome editing in a political sense? And then the second question, what is violence in the nucleus age, is really about what is the state and what is genome editing, how those two very different concepts and conceptual arrangements come to fit together on a theoretical basis, which leads me to my third part, in which I suggest a solution that is taking the Foucauldian idea of disciplinary power rather than the different iterations of biopower, and try to fit it with Rabineau's work on Giorgio Agamben's Zoe and Bios dichotomy, trying to fit in DNA in his book, French DNA, and try to make sense of this complexity by switching the idea of disciplinary power from the individual body within the society, but rather on the genetic and social outcomes of the variants, and then tracing it back to the genome editing of those variants and deciding which variants are allowed to flourish in society or not. So for the first part, the specific CRISPR controversies, what is specific to CRISPR or not. Well, first, we can have a look at the different documents that have been published. So this is a chronological representation of documents. You have three corpuses of documents. The blue ones are GMO, the purple ones are MRT, and the green ones are genome editing. So it starts in 2007 and then goes until 2020. So what I think is very interesting is that you can see that the links that you see here among documents are links that show common authors. So two documents that you see here, this is a document, for example, this is the first document, share authors with all the following, the documents, the circles, it shares a highlighted link with. So if we focus on the recent period from 2015, we see that documents that deal with genome editing also, authors that deal with genome editing also deal with MRT or with GMO debates. So this is especially seeable in France and in the US and in the UK. So for France, we've talked about GMO before in the previous sessions, it is especially clear that the anti-GMO crowd has been vocal around about genome editing, while in the UK, it's especially clear that the MRT crowd has been very vocal on the genome editing side. So this is just a way to map and to visually represent documents, co-authorship documents through time, but we can see the same data set on author base, as a network of authors, to have a clearer understanding of who are the experts and what do they share. So this is the same data set. Big circles are documents and small circles are authors. And the difference of color is the difference of corpus. So here in blue, you will have genome editing, and in green, I think we'll have MRT. And you can see that some people are the actual, the interface between the two debates. So for example, in the UK, Robin Lovell-Badge, you see George Church right there, that is much more on genome editing than on the other issues. But Robin Lovell-Badge is both present, so what they call three-parent babies is about MRT, on MRT and on genome editing. In France, we have someone like Catherine Bourguin, who is right there, that will be present both on l'Appel de Poitiers, which is the GMO, like the will by green activists to set gene-edited plants as considered gene-edited plants as GMOs, but she's also present on the heritable, so the germline editing debate. And you have a few people like that act as interfaces between debates, both through time and on a more level field, among different subjects, among different controversies. So if we move back to the content of those documents, well, most of those documents, and now I'm really focusing on those that talk about genome editing, they all mention and insist on, and the intensity of this insistence is very, very high, on the idea that they must include the public. So those are documents from 2015, before the birth of Lulu and Nana in China, but still after the publication or the rumors that a first study on human embryos would be published by a Chinese lab. So the urgent need for open discussions, the mentioning of the general public without defining it. And this is where we find the first iteration that will be summoned again and again and again in the next years of the broad societal consensus, even though, again, it's not very defined. A few years later, 2018, they're calling for broad public dialogue, and even for accounting the full range of humanity's moral imagination and insights, which is very broad, and we're not really sure how we'll manage to get there. And in the past recent years, it's grown even more wide, so all nations agreeing to proceed deliberately and with due respect to the opinions of humankind. As a political scientist, it's hard to imagine what kind of framework can get you the opinions of humankind, but you know, might want to try. And then again, meaningful public engagement and empowerment with the acknowledgement that such deliberations can be messy and challenging. Well, it so happens that those kinds of deliberations happened in France 2 years ago as a part of the effort to renew our framework for bioethics. We have laws that are specific to bioethics in France, and it had been decided that CRISPR and somatic and germline editing had to be on the menu of this new law that is still underway. It's been delayed because of the COVID, and it's still being examined by the chambers. However, what is very interesting is that there was a fully fledged public consultation that has been led with 4 pillars. So as a citizen committee of randomly selected citizens, a public debate with a series of 271 local conferences. So that's quite big. An online consultation through a dedicated website, and then auditions by the agency in charge of organizing this whole consultation, so expert auditions. Now, this agency, the Conseil National Consultatif National d'Éthique, has said in its report that no one really cared in its own experts' auditions about CRISPR and germline editing. However, this is a picture from 2016, which is pretty straightforward, but I can translate it for you. It says top GMO babies. CRISPR-Cas9, yes to therapeutic progress, no to transgenic embryo. You see the little DNA, the little, I guess it's an ICD scan, little scissors. So this is 2016, 2 years before, and this is in Paris. This is the business quarters of Paris La Défense. This is 2 years before the debate. And so when we have a look at the data, which I did, of this online consultation, this is the main 3 teams that you can see mentioned by the anonymous online participants of the debate. So the 3 main themes are large-scale eugenicism, the definition of what is a genetic anomaly, GMOs. So then again, it's more or less the same debates, but they're drawing the fears from GMOs to this new, I guess, level field of germline editing. And then the good old biosecurity, the idea of a genetic warfare that, well, most of us have already met in synthetic biology and sequencing and before. So none of this is particularly new, but it's wrong to say that it was a failure, the consultation was a failure to the extent that people were not interested. They were. It's just that apparently, and that was entirely foreseeable, activists managed to nudge the platform and have their message, a well-crafted and a well-distributed message, dominate the platform, which is in itself quite a feat. So I will move on to my second part. We're talking about, everyone seems to be talking about public engagement while calling for prior controversies to mobilize their own activists and their own pre-built political narratives. However, there is apparently a collective inability to draw a clear image of who and what is at stake with genome editing. For that, I will again use a question that has been asked by Sheila Jasanoff a year ago in a conference at Berkeley, CRISPR Consensus conference organized by the IGI, in which she asked that question rhetorically: to what extent are we designers and engineers of ourselves and of our own futures? Now, my argument here is that the main question is not the question of the extent, but it's rather, for me as a political scientist, of course, the question of the we. What is this community, and what is this group of people sharing a commun? What is this common thing that we should be talking about, made of or made from? And then how do we build that community? Here, a bit as a joke, I'm writing that nucleases are expanding the red tuna problem. You're probably aware of that text by Bruno Latour, in which he shows that how through green activism, their political campaigns, one can realize that the red tuna should not be part of my territory because I am part of his. Now, the question of the territory of the tuna is important, but the question of what is at stake with nucleases, so enzymes that are able to cut virtually any strand of DNA, and much more actually, this genotoxicity, this specific genotoxicity, is a real issue in terms of territory, because we can say this is the political space, the topological space of the tuna, and those are the policies I can enforce to make sure that it is protected or it is articulated with our own human territory. However, in the case of the nucleases, it's much harder to do. So my argument here is that a way out of this issue is to consider that a territory, not our territory or the tunas, but the nucleases. And by definition, since nucleases cut DNA, and that the definition of living organisms is being able to transmit DNA, one of the possible definitions, then what we need to take into account as a fictional legal standpoint when enacting policies is not the one of the people or the political community, the implied human community that doesn't really exist either, but rather of the biosphere. So moving on, because I don't have much time anymore, what is violence in the nuclease age? The main argument here is that we are in a very different situation that we had maybe 20 or 30 years ago when talking about gene sequencing or PCR or synthetic biology. because the categories cannot emerge. In his book, French DNA, Paul Rabinow details how people involved in genomics, bioethics, patients associations, we talked about finance and startups and even states, they all agreed that to some extent the future was at stake. Genome editing is very different to the extent that very few policymakers do believe it does the same things. A lot of my work has been interviewing those policymakers in France, in the UK, and in the US. And it so happens that most of the rules that have been taken on genome editing were taken either by judges or through writers. So not by actual laws that are dealing with the facts, but with parallel strategies to enact militant goals that are usually maintaining the pre-existing status quo itself, coming from the pre-existing controversies. Which leads me for my final 2 minutes on my proposition of solution, well, at least on a theoretical level, is to not, rather than using this biopower framework that we can try to enact from or draw from Agamben, is to go back to Foucault and try to see how we can tweak his concept of disciplinary power, selection, normalization, hierarchization, and centralization, by changing its focus from the individual body to what is at stake with genome editing according to qualitative and quantitative data. That is not the variant itself, no one really cares about the variant, but the social outcome on an individual and collective level of changing those variants. So if we focus on the outcome, we can trace it back. So the desired or undesired outcomes, such as inequality due to having access to better genes, whatever that means again. How do we trace that issue back to editing? And so how do we select the sequences that can produce or that should not produce specific outcomes? Normalization, so shifting from self-surveillance to the checking of the penetrance on a social level of an array of variants in designated spaces, political spaces, of course. Hierarchization goes from hierarchy of bodies, that is social or individual hierarchy, to the hierarchy of outcomes. So knowing that, of course, due to the complexity of the biosphere, each outcome that is marked as desirable will have effect on a series of different outcomes. And well, I guess the centralization system doesn't really change because you need to have a hierarchy to enact, to enforce issues. I don't really have time anymore, so I'll just switch the conclusion. I try to show that the current political controversies that exist around CRISPR today are not really specific to CRISPR, even though there is a level of intensity in requesting for public engagement that is much higher than what we could observe before. But at the same time, whenever those, that was the point also mentioned in previous presentations, it seems that most of these public engagement exercises didn't really succeed in getting anything passed through any kind of democratic process, which is why I'm trying to propose to argue for a shift in focus in how we build this political, or try to not even build, but even understand what is at stake, going from the variant or the tool, which has been the focus for GMOs or for actual biosecurity regulations, but starting from the social outcomes that can be traced back eventually to the desirable or undesirable outcomes. Thank you for your attention, and I'll be taking questions if there are any.